Hi everyone, welcome to our first blood bank lab. So today we're going to be learning about forward ABO typing and also typing for the big D antigen. But before we do, we're going to have a little bit of a background into the ABO system, and also before we get into that, I'd like to discuss some important terms related to our blood bank testing. So what we see on this slide, firstly, the term genotype is a term that's used to describe the actual genetic makeup of an individual. So genotype basically tells us the genes that an individual has inherited from both their mother and their father. And the relevance for us today is going to be related to their ABO blood type, okay? So genotype are the actual alleles, which we'll learn about what alleles mean in class a little bit later, that an individual inherits, and this determines their ABO blood group. When we compare this to phenotype, phenotype are observable traits that result from these inherited alleles. And basically phenotype is what the results we're going to be getting from our testing. Okay, tube testing. These are phenotypic results, so they can tell us perhaps which antigens are present on this person's red blood cell, and we can then deduce what blood group they are of the ABO system. But in some cases, it does not tell us their genetic makeup, meaning which actual alleles they inherited from mother and father to give them this phenotype. Okay, hopefully this will make sense when we go over an upcoming slide. So two other terms that are important to this conversation are heterozygous and homozygous. So you all know that the prefix hetero means different. So if an individual inherits two different alleles from their parents, they're called heterozygous. They would be called homozygous if they inherited the same exact allele from each parent. Okay, so they have two alleles that are the same. So this chart kind of breaks phenotype and genotype down by blood type. Okay, so on the left-hand side here, we have the four possible ABO blood types or blood groups, and basically, we're going to look at possible genotypes for each group. So when we have an individual who is blood type A, they have the A antigen present on their red blood cells. But as far as their genotype, it could be one of two different patterns of inheritance for this individual. So they could be homozygous, and they could have inherited an A allele from mom and an A allele from dad. That would give them blood type A. This is a phenotype. Or they could have inherited a heterozygous combination. And here we see an A allele from one parent and an O allele from the other. So when we look at these blood group antigens, they are inherited codominantly. So basically what it means is that whichever antigens are inherited are expressed. One is not dominant over the other. However, when someone inherits an O allele, it doesn't code for a functional protein, and so it's kind of like a zero. Okay, it just doesn't contribute any kind of antigen to the red blood cells. Okay, so basically type A could have a genotype of AA homozygous or AO heterozygous. And so we would need to look at family studies to determine the genotype or do molecular studies. And so in lecture 4, when we talk about genetics and inheritance patterns, we'll do some examples. Sometimes knowing what your mother or father or siblings are, you can deduce the genotype, but not always. So let's talk a little bit more about blood type A, and this should actually say can receive red blood cells from, okay? So packed red blood cells are the most common type of product that are transfused. When we have an A individual, you can always get your same type. Okay, so you're going to see that throughout. Okay, your type identical is always good. So we can always give them type A. But when we're talking about red blood cells, individuals that are type O are the universal red blood cell donor. Okay, so anyone can get O red cells. Moving on to type B, okay, they have the B antigen on their red blood cells, and this is their phenotype. The genotype, once again, could be homozygous, a B allele from both mother and father, or they could be heterozygous, inheriting a B allele from one parent and the O from the other. Okay, they're still going to be B regardless, okay, because remember this one is silent, but they still have the B, it's still going to be expressed. So individuals who are type B can receive red blood cells from their same type B or O, which is the universal red cell donor. The rarest blood group that we see is type AB, and so this clearly shows how ABO antigens are expressed codominantly, because this person has, people that are AB have both the A and B antigen. This is a unique case where the phenotype has to be the same as the genotype, because they would need to have gotten an A from one parent and a B from the other in order to express both of these antigens. So with type AB, phenotype and genotype are the same. When it comes to looking at what types of red blood cells this person can be transfused with, although they're the rarest type, interestingly, they can get red blood cells from any of the types. They can get from type A because they have A antigen. They can get from type B because they have the B antigen. They could get from AB, or remember, O is the universal red cell donor. And lastly, most common blood group among all races, ethnic backgrounds is type O. And this is another instance where the phenotype is the same as the genotype. In order to be type O, you have to be homozygous for O alleles, meaning an O from your mother and an O from your father. That's the only combination that will result in blood group O. And so when we look at the type of red cells that O individuals can receive, you will notice that they can only get O. Okay. So although O is the universal red cell donor, O must receive O. Okay, it can't get AB or AB, or they will have a transfusion reaction. Okay, so that's just a little bit of background about phenotype and genotype. So we're going to go over this in class, probably in lecture 5 as well, but I thought I would just talk a little bit about it now. When we're referring to these ABO alleles, okay, I already mentioned to you guys that they're codominant. What I didn't mention to you all is that they actually code for enzymes that transfer a certain sugar to form the antigen. Okay, so it's not like they just pop up there and it's an antigen. It actually is a transferase enzyme that places a sugar on a building block, and that's what makes the antigens. And so codominant, remember, means that both antigens will be expressed if both alleles are inherited. Getting back to the O allele, okay, so it does not code for a functional enzyme. So O just happens to be a defunct allele. It doesn't code for a transferase enzyme that transfers a sugar, and so there basically is no antigen produced. So this picture I thought would be very helpful, just to kind of give you a view of what's actually happening on the red cell membrane. And so remember, these antigens, we like to think of them as being on the surface of the red cells, but they're basically embedded into the cell membrane, and most often they're glycoproteins, and so they are structurally part of the red cell membrane. As we go through a little bit later and we learn of other blood group systems, we will find that some antigens of some systems how can I put it? Let me put it a different way. Scientists have determined the function of antigens of other systems. And so meaning that these proteins or glycoproteins that are embedded in the membrane aren't just there haphazardly, they actually do serve a purpose for the cell, okay? And so here there's a nice picture. We can see the A antigen on A red cells. The B looks different, looks like a lollipop on the B. And then when we look at AB, we see it has, you know, both lollipop and the arrow, if you want to call it that, okay? And then O, you see nothing, okay? Sometimes seeing a picture helps. So this man is basically the father of blood banking. Carl Landsteiner. He looks pretty stern there, doesn't he? But thank God for him, because I'll tell you what, without him, transfusion medicine would not be what it is today. So basically, Landsteiner discovered in the 1900s that people that have certain ABO groups also have naturally occurring antibodies to the antigens that are not present on their own red blood cells. And this is known as Landsteiner's rule, okay? And we're going to look at this more carefully in a minute, but it's very important to know this. So they're naturally occurring, meaning you don't have to have a transfusion or be pregnant with a fetus with different antigens than your own to make these antibodies. They are just naturally occurring. And we have antibodies to the antigens that we lack, okay? So to put this in another way, an individual would never have an ABO antibody to an antigen that is present on their own red blood cells. So if you happen to be type B, you would never have an anti-B in your system because you would be agglutinating your own red blood cells. So Landsteiner figured this out. And thank goodness that he did, because you know, around the turn of the century, people were, or I should say, doctors were experimenting with transfusions of blood, and sometimes it worked, and sometimes people died within minutes or seconds, and it's because they got a blood group that wasn't the same as their own, that was incompatible, and it was really haphazard. So him discovering this really improved the outcome of transfusions, and many more people lived as a result. So this is another chart that basically compares all of the blood groups, which antigens are present on the red cells, if that may be, and then also naturally occurring antibodies that an individual would have. Okay, so we're just going to basically review this. So when we have type A, okay, they're called type A because they have that A antigen, okay, and since they have type A antigen, they would only have an antibody to the antigen they lack. They do not have B, so they would have anti-B. Okay, so A antigen with anti-B. When an individual is type B, they have the B antigen on their red cells. What do they lack? They lack A antigen, so they're going to have anti-A. When we look at the unique individuals that are type AB, they have both antigens on their red cells, and since they have both, they don't have any naturally occurring antibodies. They wouldn't have anti-A or anti-B because they have both of these antigens. And then finally, group O, they have neither A nor B antigen, so they're going to have both anti-A and anti-B in their plasma. So hopefully that makes sense to everyone. This is important to get down because when we talk about forward typing, okay, we use known antisera to determine the antigens that are present, if any, on the red cells of the patient. When we do reverse typing in lab 2, we use known reagent red cells to determine which antibodies, if any, the patient has in their plasma. So we use both. And blood bank testing is unique because it's one area of the lab where the red cells are separated from the serum and the plasma, and each is tested separately. Okay, so this is just another chart that basically kind of wraps everything up. Okay, so we have our blood group phenotypes here, antigens that are present, hopefully that makes sense, antibodies we would expect, so you have A antigen, you have anti-B. B antigen, anti-A. AB, both antigens, no antibodies. O, no antigens, both antibodies. And then possible genotypes, which we talked about previously. So just remember with type A and B, there can be more than one choice. Okay, they can be either homozygous or heterozygous. When we look at A, B, and O, there's only really one choice. So specimens for blood bank testing, most labs use EDTA anticoagulated blood. Most of them use the pink EDTA tube. I'm sorry, I could not find a good picture on the internet, so you're going to see the lavender. They would separate this plasma from the red cells. The red cells would stay in the original tube. The plasma would go into an aliquot tube that has the same label on it, okay? And they're rubber banded together as a sample set because both are tested in blood banking. Plain serum tubes without gel can also be used. So a plain red top with no gel could be used. We would centrifuge it, separate the serum from the cells in the clot. If you're in a lab that uses serum tubes, it's just a little bit more challenging because to get the red cells out of the tube, you have to mash the clot with some sticks to liberate some of the cells. So most labs don't want to be bothered with that, and they prefer the EDTA, which the cells in the bottom, when they're separated, we could just take a pipette and take a few drops of them, for testing. Much easier. Hemolyzed samples in blood bank are not acceptable. And so if I look over here, maybe that one's not centrifuged. I was hoping I could see one that is pink or red. The reason being is when we're doing blood bank testing, we're looking for agglutination reactions of immune complexes forming. However, if hemolysis results in our test system, that also indicates a positive reaction. It could mean that complement was activated, causing lysis of some of the red cells in our test system. And so if we start off with a hemolyzed sample, it's not going to be good because we won't be able to detect any changes. So they're canceled, would have to be redrawn. So let's outline our classic tube testing for ABO. When we're doing forward grouping, as I said, we use known antisera. Antiserum means antibody, okay? And so these antisera are directed against the A and B antigens. There's actually a combo reagent called anti-AB that would react with both A and B antigens. And we're using these known antisera to determine which antigens are present on the patient's red cells. So the idea is, let's use the example of anti-A. When we put anti-A in a test tube with red cells from a patient who's type A, that anti-A says, hey, there's my target, there's A antigen, and it links up and forms an immune complex, and we see agglutination in our tube, okay? RHD testing, the RH system has quite a few antigens in it, but by far the most important is the big D. And so when we're testing for this big D antigen, there is a separate antiserum known as anti-D. It's an antibody directed against the D antigen. And so when an individual's red cells have the D antigen, it's going to agglutinate when we add anti-D to them. The D antigen is what confers Rh positivity to an individual. So if the cells do agglutinate with anti-D, it means this person is Rh positive. If we get a negative reaction with no agglutination, looks like that person is most likely Rh negative, but sometimes we have to do additional testing to be sure. So that's our forward typing. Okay, so with forward typing, remember, the known is antisera. The unknown is which antigens does our patient have on their red cells. So we're using a known to help figure out an unknown. When we get into reverse grouping, and we'll be looking more carefully at reverse grouping in lab 2, we're using known reagent red blood cells. They come from a company in vials. So we have reagent A cells and B cells, and we know which antigens they have, okay? We're using these cells to determine which antibodies, okay, the person has in their serum or plasma, which ABO antibodies. So remember, they're naturally occurring. And so we do this as a double check on what type we get for the forward grouping. Okay, so it's just basically a double check. So in blood bank, the reagents used for ABO typing are pigmented intentionally to reduce mistakes. So anti-A is always going to be blue, no matter where you are, no matter what company's reagent you buy. Anti-B is always going to be yellow, and then most of the other ones are clear. So here you see a vial of anti-D, and that is just clear. Okay, so when we're getting ready to do testing on our patient's red cells, we don't just test them straight out of the original EDTA tube. We have to prepare a certain concentration of the red cells, and the one that is typically used is 2 to 5% for tube testing. Okay, so the reason being, if I was just going to take a drop of those straight red cells out of the original tube, those cells would have a lot of antigen on them, and they may overwhelm my antisera when I'm doing testing. So if you had a chance to listen to some of the lectures, you know about the precipitation curve, how we want, or we're going to be able to visualize agglutination in that zone of equivalence. Okay, so preparing red cells to a 2 to 5% is optimal for our tube testing. So how do we do this? We take a few drops of red cells from the original EDTA tube and place the drops in a newly labeled test tube. Okay, and then we're going to use physiologic saline. We fill the tube with physiologic saline. We cover it with parafilm. We mix it very gently, and we're going to centrifuge for 1 minute at 3,500 RPMs to wash the cells. Okay, so washing the cells is important because it's going to remove any kind of unbound proteins that are present in that sample. And if you have fresh specimens, you're going to wash them once, and the wash is going to look pretty good. It's not going to have a lot of pink or red color to it, and that's good enough. But washing, just remember the purpose always is just to remove unbound proteins and complement that might be in the tube. So we're going to decant that wash solution into a biohazard container, get rid of it. We just tilt the tube without hesitating and pour it off, so to speak. And the red cells, we're still going to have some red cells in the bottom of that tube. And now to these washed cells, we're going to add more saline, but we're not going to fill it all the way. We're going to fill it halfway, maybe to 2/3, and we're going to cover it with parafilm and mix it and compare that tube of red cells to what our reagent red cells look like. We hold the bottle and the tube and we compare them with the lamp to see if the turbidity is similar. And so it's an eyeball method to get that 2 to 5% solution. Okay? Because these come prepared as 2 to 5%. So that's how we make our suspensions. And that's what we're going to use for testing. So the actual forward typing procedure for tube, I have a chart here from lab. And you know, even though this course is being taught completely virtually, hopefully in the fall when we meet face to face, we will have 1 or 2 labs just to make sure everybody knows how to do the testing. I think that's important. So we're going to label 4 tubes. I'm sorry, I have a mistake here. FA, FB, and if we're doing RHD, it would be RHD and our patient control. Okay? We always want to add our serum source first, whether it's an anti-serum or from the patient. Okay, we always want to add that first. And so, you know, we can see here that, oh, okay, I have FAB in here. This is the anti-AB reagent, and not all labs will do that testing with anti-AB. Sometimes they save that as a double check if there are some discrepancies. So we add our serum source first, which in forward typing, remember, is an anti-serum, the known. So we're going to have our 4 tubes. Okay. So first one, we're going to add a drop of anti-A into the FAs, into the FB. And we would have the patient's identification number, lab number on each one of these tubes as well. FB tubes are going to get 1 drop of anti-B. FAB would get 1 drop of the combo anti-AB, and then the RHD gets 1 drop of the anti-D. And then we're going to add 1 drop of that 2 to 5% red cell suspension that we made into each one of these tubes. Okay, we give them a little mix, and we put them in our centrifuge and we balance them. So tube across from tube. And since there's only 2 drops of liquid in each one of these tubes, there's no need to cover them with parafilm. There's really no splashing that happens. And here's the difference. We're going to centrifuge at 3,500 RPMs, but only for 15 seconds. So just remember, when you're centrifuging to wash cells, it's a longer time. You want to get rid of the dirt, right? The dirty laundry, wash longer, that's 1 minute. When we're centrifuging to read a reaction, it's only 15 seconds centrifugation. We're going to take our tubes out of the centrifuge and we're going to put them back in our test tube rack in order before we start reading. I think it's really bad technique to take the tube out and read it as you're taking it out, because you might not be taking them out in order, and that's when mistakes are made. And in blood banking, there is really 0 room for mistakes, because mistyping someone could lead to them getting an incompatible blood type and death within minutes. So this is a picture of a rack that I set up last year. And so rack organization is critical as well. And I suggest you organize your rack the same way every time. That way you don't make mistakes. So here you're going to see, this is a good example. I have patient 24, and you can see that was a lavender EDTA. This is patient number 24's red cells, and over here is their plasma. So you can see how they're separated. I have a row that I separated between the samples, and then my next patient was 37, and you can see these are the red cells in a pink top, and here is 37's plasma. And then what I'm going to do is, you can see 24, I have the 3 to 5% red cell suspension, or 2 to 5%, really could be either one. 37's 2 to 5% red cell suspension, and then FA2 for 24, FA2 for 37. I could test more. All of the FAs would be in a column going this way. Okay. The FB tubes, 24 and 37. You can see I would have all of my FB tubes in the same column. And then the FAB, if I'm doing that test, they would all be in the same column. And then the D tube, they would all be in the same column. Okay. And so by organizing myself that way, I'm going to be less likely to make mistakes. Okay, so hopefully that makes sense. Okay, and so once you have everything set up, the tubes labeled and organized, it's really easy to do the testing, because what I'm going to do is, remember, we had our serum source first, all of these FA tubes. I'm going to take my anti-A reagent, my blue reagent, 1 drop here, 1 drop here. If I had more patients, I could be just doing 1 drop down the road, right? 1 drop of the yellow anti-B into these, 1 drop of the anti-AB into these, 1 drop of the D into these, okay? And so it makes it easy. So anti-sera go that way. Then second thing I'm going to do is take 24's 3 to 5%, 1 drop into all 4 tubes. 37's 1 drop of their 2 to 5% red cells this way. Okay, give them a shake, centrifuge them. There's an example of one of our centrifuges. You can see I would turn it on to 15 seconds, take them out, put my tubes back in order before I read. So when we're reading the reactions, we have special lamps that we use in blood banks. So this is actually a lamp that has a mirror on the bottom. And we want to read 1 tube at a time. Okay, this is only 1 tube. And so when you pick up the tube from the test tube, you want to have the button of the cells like facing in your mirror so that you can see it magnified when you go to read it. Okay, and you're just going to gently mix it once or twice, and then look in your mirror to see what happens. And so we can see here how there are 2 big clumps of red cells. that are present in this one, and that's going to be important for us to grade our reaction. Oftentimes in lab, students will take the tube and they'll like shake it like 10 or 12 times before they even read it, and I say that's really bad practice, because if you're shaking it a whole bunch, you could shake away positive reactions. Okay, so you just want to shake it once or twice and look. If nothing's coming off, shake it once or twice again and look until the button is completely off the bottom of the tube. So when you have positives, they're going to show up more quickly. When you have a negative, you may have to keep mixing it until, you know, everything's completely off the bottom of the tube, and a negative is going to be completely smooth red cell suspension. So in blood bank, all positive reactions are graded. And as I said before, please, no overmixing of the button. Mix and read, etc. until it's off the tube. So I started with negative. This will take the longest. Negative reaction we see down here is a smooth suspension of our red cells in the mirror with no clumping. Okay, it's a cloudy, turbid solution of the red cells. 4+, we have one solid clump that comes off the tube. 3+, 2 or 3 large agglutinates, and the background is clear. Okay, you can see how it's clear. 2+, we're going to have multiple clumps. They're getting smaller, but the background liquid is still clear. And then when we get to 1+, the agglutinates might be getting smaller, but the background is no longer clear. It's starting to look cloudy. So you can see it's not clear like the 2+ anymore. It's starting to look cloudy, so that's a 1+ reaction. Okay, and that finishes that. So what you should do next is there are a couple of videos to watch where people are performing the washing steps and setting up the tubes and reading them. And after that, there will be a lab activity for you to interpret.